Okay, yeah, I think we can start. Hello, everyone. Good evening, good morning, good afternoon, wherever you are. Welcome to the first webinar in which we are presenting the GRA Feed Additives Flagship Project results, which are the technical guidelines for methane-reducing feed additives. Thank you for joining the webinar. As you know, we have recently published a special issue in Journal of Dairy Science with 6 papers, including different aspects to consider when developing and implementing the use of methane-reducing feed additives. Next slide, please. And I don't think in this group, maybe I need to emphasize too much about the importance of feed additives. We have a very recent meta-analysis and reports showing that feed additives are probably the most effective, or one of the most effective strategies to reduce enteric methane emissions. Extensive research has been conducted in the last decades. But also recent reports that, like the one published by Hegarty et al. Recently, also have shown that despite that extensive research and lots of effort put in by the academic community, the industry, the number of feed additives are available in the market is very limited. And therefore, more efforts need to be put in place to further develop this discipline. Next one, please. So after different discussions within the Global Research Alliance and the Feed and Nutrition Network, the Global Research Alliance decided to launch and support a project which main goal was to try to facilitate development and the use of feed additives by putting together a global network of experts that could first share knowledge, discuss, and create some kind of document that could be used as guidelines and recommendations for both the academy and the industry to develop and apply feed additives. And this, we recognize that was not a very easy task. So first, we had to not only get the support from the GRA, but also trying to recruit people that could help us to undertake this goal. Next, please. And this was done by a core team of people that I have the privilege to work with. Andre Banning from Bargaining and Research and myself have been coordinating this project together with Florencia Garcia from Argentina. And we have put together a group of world experts in the different aspects needed to provide these technical guidelines and recommendation, and you have the names in the slide, and you will see some presentations by some of them in the next minutes. There have been more people, as we will see later, but without the commitment and the care of these global experts, this project wouldn't have been possible. Next, please. So in total, 60 researchers participated in this project from 23 countries and 46 institutions. So a real global international effort. Next, please. To put together these 6 papers, and this is just a graph trying to illustrate the different aspects and the different steps covered in the guidelines. As you can see, we have on the left-hand side the different aspects related to the development, and then on the right, those related to the implementation. In regards to the development, of course, first, the candidates for developing feed additives need to be identified using different approaches, and then an initial screening need to be done. And then further research conducted either in vivo or using modeling approaches. and eventually trying to uncover the mode of action. This is all about the development, but we also recognized that there were other aspects that were clearly limiting the implementation of feed additives. These are related to the regulation and the registration processes, and there is a paper led by Juan Tricarico addressing all those issues using the regulatory landscape in different regions of the world, and eventually also another manuscript trying to describe how we can account for the reductions in carbon emissions when these feed additives need to be used, and how to do it at different levels, farm or national levels for inventories. So this is just a summary of the whole view of the technical guidelines. And now, if you pass to the next slide, please, Flor. Yeah, this is the program or the order of the different items of this first webinar. We're going to have this webinar, and then in 2 days' time, we have going to have another one. So first, the 6 papers will be presented in a pre-recorded presentation, so just to make sure that we don't take too much time for the presentations. And my colleague Andre Banning will lead the questions and answers. So from now, when the presentation starts, you can use the questions and answer option, so you can paste your questions there. We're going to try to discuss all of them, and if we don't have time during this webinar, then we will also try to address the questions that remained not addressed, and then send a link with the answers or comments to all of you. So next one, Flor. Yeah, this is the webinar it's going to happen in 2 days. So again, any questions, please paste them from now in the questions and answer option. And I think we are, yeah, if you pass to the next one. Yeah, again, thank you very much. So yeah, this is what we're going to do now. We're going to go from 1 to 6. So the first one is a paper led by Zoe Durmik and Everdoing on the identification of new feed additives and how to conduct initial screening in the lab using different approaches. And I think we are ready to go.
Yes, good morning from Australia. My name is Zoe Durmic, and this is the first paper where we will introduce the initial steps to discovery and testing of antimethanogenic compounds. As you can see, we had quite a lineup of expertise with Ever as a co-lead in this group, people of different expertise and all around the world from different disciplines. In the next couple of minutes, we will reveal what are the steps to discover, evaluate, and select candidates, followed by some recommendations on the process and the methodology. As you can see, it's a quite complex process where we start by searching for candidates, then we source, identify, and test. And finally, we evaluate and select the ones to progress to further testing. In general, there are 2 methodological approaches to search and identify: empirical and mechanistics. In this empirical approach, it is well known and broadly used, and there are about 3,000 peer-reviewed papers published. So basically, we source for information in the literature to identify candidates, and then we go out in the field where we collect parent material. And then from this material, we then extract and we test the material in vitro assays. And in some instances, we can progress and actually through rounds of fractionation, we can identify, isolate and identify the single compound. The advantages of such approach is that we can assess various products in a relatively short time and in a controlled laboratory setting. We can also examine doses, interactions, toxicity, selectivity of compounds. And as I said, it can be coupled with isolation of active compounds. Now this other one, mechanistic approach, is relatively novel one, where we rely on and use computational tools and online platforms, basically to target methanogen unique cellular processes. So what does it mean? We search for the compounds that can potentially interfere with these cellular processes, and then we can synthesize those target compounds and then confirm their test and confirm their activity again in vitro. The advantages of this approach is it is fast and cost effective, it has unlimited potential, there is no need for specialized equipment, and some compounds can be synthesized, so there is no need for parent material. Once we have selected compounds, we progress to evaluation of candidates, which can happen at subcellular level when we're testing the effect on biological processes, for example, inhibition of methanogen enzymes, commonly in these plate assays. The next one is a possible test on methanogen, the whole methanogen cell, where we can actually rule out cidal versus static via testing in a pure culture. And finally, we can test in a mixed rumen population, where we test the effect on whole microbial community in batch or continuous culture. Finally, we have to put some filters on the selection of candidates, and in that respect, we look for some positive attributes, such as if they're readily available, if they're active at low or achievable doses, if they can promote rumen microbes, or if they're stable in their environment, also if they have persistency of the effect. Likewise, we try to rule out some negative attributes, such as limited availability, high or repeated doses required, if they're volatile, if they're perhaps susceptible to degradation or adaptation by rumen microbes. And finally, we summarize some of the recommendations that when you're in this first phase of development. So in terms of discovery, try to identify the specific targets and candidates with specific bioactive effects. Confirm bioavailability of these selected candidates. In terms of evaluation, try to select testing that matches your aims and capabilities. And also try to combine assays and methods rather than using a single one. And finally, select candidates with specific rather than general effect. Consider interactions, doses, interactions with diet, variability, toxicity. Take into account the effect on animal, human, and environment. And finally, our final recommendation, consider and follow published and verified technical recommendations like this one. Thank you.
Thank you, Zoe, for a very clear presentation. Of course, there are more details in the manuscript about the recommendations that you can take a look at. Now we're going to move to the second presentation made by Alex Christoph on assessing the effectiveness of the different candidates at animal level.
Hello, my name is Alex Christoph. I'm with the Pennsylvania State University in the United States, and I will present the second paper in the special issue of Journal of Dairy Science titled Recommendations for Testing Enteric Methane Mitigating Feed Additives in Ruminant Studies. This is the team that put this work together. As you can see, very international team, 25 or so scientists from all over the world. Peter Wund from Denmark and myself led this effort. The main message of this paper is evaluation of effectiveness of methane-reducing feed additives requires rigorous, repeatable, and scientifically based experiments with live animals. This is the reason we got this team together and wrote this work. Some of the topics covered in the paper are shown on this slide. We talk about experimental designs, basal and experimental diets effects, combining methane mitigation strategies, methane measurements techniques and protocols, other processes to monitor and measure, animal health, and animal product quality. We don't have time to spend on each of these topics. I will summarize very briefly some of the main points. In terms of experimental design, we would be looking at pre-experimental power analysis, post-experimental outlier analysis. We emphasize the importance of the design of the experiment in relation to the objectives, and very importantly, the need for long-term studies with different ruminant species and production systems, and particularly dietary conditions, forage quality, to have a full spectrum of the effectiveness of these feed additives. We also discussed residual effects. We discussed combination of feed additives, nutritional practices. They may have associative effects, they may have additivity, they may have negative effects as well, and how these strategies may or may not be combined with non-nutritional strategies, and whether there will be a combined effect on methane emissions. We discussed in length the different techniques for measuring enteric emissions. The conclusion is that at this point we have 3 established and acceptable techniques: respiration chambers, green feed system, and the SF6 technique. Others can be used under limited conditions, and these are specified in the paper. Some of those necessary or useful measurements, in addition to gas emissions, have been also discussed. Number 1 is feed intake, dry mat intake. We also will recommend looking at fermentation effects, profile on the rumen microbes. Animal performance, of course, is very important. Total tract digestibility, hydrogen emissions, particularly with these strong inhibitors that are being developed and have been tested in recent years. It is very important, of course, to have healthy animals involved in these experiments, and also monitor long-term effects on animal health of the feed additives. Reproduction also is very important, but this cannot be done in small and short-term experiments. Where it's possible, this should be followed with a larger trial once the feed additive is established or tested in a different experimental design and has been proven to be effective. The overall conclusion from this work is that enteric methane mitigation claims should not be made until efficacy of the anti-methanogenic feed additive is confirmed in animal studies designed and conducted according to the guidelines provided in this paper. Thank you for your attention.
Thank you, Alex. I'm aware of the complexity of putting together this manuscript, not only because it involved a lot of colleagues, but also a range of topics and make the compilation quite complex, and that's much appreciated. Can I remind participants, if you have any questions, you can put them now in the questions and answer section. And now we're moving to the third manuscript led by Ermias Kebreb and Jan Dijkstra on modeling animal at farm level.
Hello, everyone. It's my pleasure to present key insights from our recent paper published in the Journal of Dairy Science on modeling the impact of feed additives on enteric methane emissions. This study provides recommendations on how to best model these mitigation strategies and ensure accurate quantification at farm, regional, and national levels. If you are interested, you can access the paper using this QR code on this page. The team was led by Dr. Jan Dijkstra and myself, with overall guidance from Drs. Yanis Ruiz and Banning. The co-authors for this paper include Drs. Garcia, Congio, Ellis, Eugene, New, and Bibart. The manipulation of rumen fermentation through the use of anti-methanogenic feed additives had been receiving quite a significant attention over the last few years, up to a decade. These additives, often administered in small quantities, hold the potential to significantly reduce rumen methanogenesis. Several anti-methanogenic feed additives have been investigated for their effect on methane emissions, showing considerable variation in their effectiveness. These include differences depending on the target animal. Could be beef versus dairy, physiological status such as growing versus lactating, feed management system, confinement versus grazing, diet characteristics, including forage to concentrate ratio, which vary locally and globally. There is a need to develop models that can help to understand and predict the effect of anti-methanogenic feed additives on enteric methane emissions across various production systems and diets, reducing the reliance on costly experimental studies. The aspects addressed in this paper include types of models, synergies and trade-offs of combining anti-methanogenic feed additives and model application. There are three main approaches to modeling enteric methane emissions. This include empirical, mechanistic, and hybrid models. Empirical models, including meta-analysis, regression, and machine learning, rely on existing data to generate predictions. Mechanistic models describe biological pathways and how feed additives interact with the rumen microbiome, while hybrid models combine different elements of both empirical and mechanistic approaches, improving prediction accuracy while also maintaining biological relevance. Choosing the right model depends on the availability of data, the level of detail that's required, and also the specific research or policy objectives. There were several recommendations included in the paper. To give you the highlights, these are related to model selection based on data and objectives, integration with national greenhouse gas inventories, accounting for additive supply frequency, quantifying long-term efficacy, data quality and transparency, and leveraging machine learning and hybrid approaches, and comprehensive assessments. In the next few slides, we present the key recommendations in more detail. each modeling approach has its strengths and limitations. Empirical models provide data-driven predictions, but they lack insight into the underlying biological processes. Mechanistic models capture biological pathways, interactions, but require extensive validation and system knowledge. Hybrid models offer the best of both worlds by integrating empirical data with mechanistic insights, leading to more reliable and robust predictions. To improve methane mitigation strategies, we need to align modeling approaches with real-world applications, ensuring practical implementation at the farm and national inventory levels. Selecting the appropriate model depends on multiple factors. First, the availability and quality of data significantly impact the model's predictive accuracy. Second, the level of detail that's required for rumen fermentation modeling must be considered. And third, compatibility with farm-level assessment and national greenhouse gas inventories is really important for ensuring the alignment with broader sustainability goals. And models must integrate accurate methane emission estimates at the animal level to properly quantify trade-offs and synergies with other mitigation strategies. In order to enhance the integration of feed additive modeling with national greenhouse gas inventories, standardized approaches must be adopted. The key consideration is the frequency of additive supply, particularly in pasture-based systems where pulse dose effects can alter methane mitigation outcomes. Instead of applying generalized predictions, separate models should be developed to account for transient effects in pasture-based systems. And importantly, we need to quantify the adaptation potential of feed additives in order to assess their long-term efficacy and sustainability. In conclusion, future research must prioritize high-quality, peer-reviewed data to enhance model accuracy. Important factors to consider include what's the additive dosage, what is the delivery method, and the transient effects, particularly in pasture systems where consistent intake is challenging. Machine learning and hybrid modeling approaches can also further refine predictive power, but comprehensive assessments must also account for trade-offs and synergies with other greenhouse gases. Strengthening the connection between research, modeling, and policy will ensure that mitigation strategies are both scientifically sound and practically feasible. Thank you for your time, and we look forward to discussing the paper with you.
Thank you very much, Ermias, for presenting this work that with probably some of the animal scientists are also familiar with, but equally necessary for developing feed additives. So now we move on to another aspect, very important in the process of developing feed additives, which is trying to understand how they work using a range of different methodologies. And this paper has been led by Alejandro Belanche and Emilio Ungerfeld.
The fourth paper of the series, which you can access using this QR code, is entitled A Guideline to Uncover the Mode of Action of Anti-Methanogenic Feed Additives for Ruminants. And first of all, a big thanks to all of the authors of this paper. We classify feed additives inhibiting methane production in the rumen into 4 different types according to the mode of action. First of all, there is those that lower hydrogen production in the rumen. And hydrogen is the main fuel that donates reducing power to methane formation. So you lower hydrogen, you lower methane. A second approach is to inhibit methanogens directly. For instance, you could inhibit enzymes of the pathway of methanogenesis using the nitrooxide propanol, or methane halogenated analogues like bromoform contained in the red algae Asparagopsis. A third approach is to use alternative electron acceptors, and those could be inorganic, like nitrate and sulfate, or organic, or pathways like homocytogenesis. And finally, a fourth approach is methane oxidation using methanotrophic bacteria and archaea. Some important concepts. This research is expensive and laborious, so we only recommend to conduct this research on feed additives inhibiting rumen methanogenesis with a proven and established effectiveness to inhibit methane production in vitro and in vivo, and no adverse effect on animal production and health. And secondly, we want to make a difference between the mode of action, which refers to the effect of the additive on specific microorganisms, the microbial ecosystem, and the host animal, and the mechanism of action, referring to the molecular interaction between a certain compound and a microbial cell. So which are the main research recommendations in this paper? From the microbiology perspective, the central question is which microbes are targeted by the feed additive. And initially, we can describe changes in the microbial community using techniques like qPCR and sequencing. However, that won't allow us to discern between direct and indirect effects, which we need to establish direct effects using pure cultures to study toxicity of the feed additive on certain microbes, and then evaluate whether the target microbes can be or become more resistant to or develop resistance to the feed additive. And finally, evaluate the possibility of potential undesired side effects on non-target microbes. At the cell and molecular biochemistry perspective, in case of feed additives from natural origin that contain a blend of compounds, we first need to identify the active compound or compounds, and then both in case of feed additives from natural origin and pure chemicals, hypothesize a mechanism of action based on the chemical composition and molecular structure of the active compound, and then use an array of different chemistry and biochemical and microbiological techniques to elucidate the mechanism of action of the active compound. At the microbial fermentation and ecosystem perspective, we need to characterize changes that occur in fermentation product, and particularly accumulation of intermediates is quite important to understand which pathways are inhibited. We need in vivo confirmation of initial research conducted in vitro, and all of the above is going to be complemented with omics techniques like genomics, metagenomics, metatranscriptomics, metaproteomics, metabolomics. And finally, studying how the feed additive is metabolized in the rumen involves a variety of techniques, and if the metabolites of the feed additive are naturally present in the rumen, isotopic labeling may be necessary. From the whole animal metabolism perspective, if we observe decreases in dry matter intake, we should evaluate the possibility of palatability issues by comparing the administration of the feed additive mixed in the feed and through the rumen cannula, and evaluate the possibility of inflammation of the digestive tract using cell cultures, histological examination, and blood inflammatory indicators. And we need to characterize the metabolism of the feed additive post-absorption study its accumulation in different animal products and organs, and evaluate the potential toxicity and mutagenic effects of the additive and its active compounds. The cross-cutting perspective studies how factors like type of diet and type of animal and other factors interact with the efficacy of the feed additive. And in order to do that, we need a thorough and detailed explanation of the methods in scientific articles. We recommend to monitor the concentration of active compounds in the rumen to understand the variation in effectiveness of the feed additive throughout the day. And finally, it's important to understand the individual mode of action of each feed additive in order to seek potential synergistic and additive effects. And thank you very much for your attention.
Thank you very much, Emilio. Again, another massive piece of work, considering the range of different methodologies and approaches that can be used to uncover and describe the mode of action. So lots of potential questions there. We can see that there are questions coming now. Some the direct questions have been answered, and you can see that in the Q&A section. Those that have not been answered, they are being listed, so we can open the discussion led by Andre Banning later on. Right, now we move on to the second group of papers, two papers dealing with the aspect of implementation. So the first one, something that, again, animal scientists in general probably are not that familiar with, which is the process. First, the regulatory landscape. What are the differences and the commonalities in the regulation in different regions of the world, and what aspects need to be considered when addressing that last stage, when it goes into that stage or before, when the experiments need to be conducted. So Juan is going to present our work right now.
Hello, my name is Juan Tricarico, and I am going to present on regulatory frameworks and scientific evidence required for feed additive authorization. You can access the article using the QR code on the screen. Before I start, I want to recognize all the individuals that contributed to this review article. I also need to clarify that I am an employee of Dairy Management Inc. and that the scientific and professional views expressed in this article are mine and do not reflect the views or opinions of my employer. This review article had three main objectives. The first was to outline regulations and evidence requirements to obtain authorization to mitigate enteric methane with feed additives. 7 jurisdictions were examined as illustrative examples. These outlines are intended for applicants and scientists to understand evidence requirements and facilitate compliance with regulations. The second objective was to identify consequential differences and similarities between these jurisdictions. These are useful to improve harmonization among global regulatory entities. The final objective was to provide recommendations for applicants and the scientists evaluating feed additives for approval. Before presenting the main article findings, I want to share with you the meaning of few commonly used terms. Starting at the top, regulation refers to a set of rules. Their objectives are to protect animal health, promote food safety, and prevent misleading claims and unsafe practices. These rules are legally binding and enforced by regulatory authorities within each jurisdiction. Regulatory status, also commonly referred to as legal classification, are feed ingredient definitions set by the authorities. The criteria vary by jurisdiction. For example, some jurisdictions rely on desired effect, while others on mode of action to legally classify the additives. Next, the term registration is used in some jurisdictions to refer to the authorization procedures established by the authorities. These procedures define the evidence requirements for applicants pursuing authorization to market the feed additives and are established based on the additive's regulatory status. Conditions of use are the practical aspects of feed additive administration. These include active ingredients, doses, mixing conditions, feeding frequency, and withdrawal periods if appropriate. And finally, a very important term is intended use. It refers to the specific purpose for which the additive is designed and recommended for inclusion in target animal feeds, for example, methane mitigation in lactating dairy cow diets. As I said, intended use is very important. Why is this? The answer is simply because all authorizations are granted based on intended use. Applicants are required to submit a clear definition of the intended use, which determines the additive's legal classification, the scope of evaluation, and the evidence needed for approval. This includes evidence on effectiveness for the claimed purpose, target animal and human food safety, and environmental impact. Finally, conditions of use also need to be clearly communicated on all product labels. There are two additional considerations stemming from the intended use concept. First, many substances are novel or represent a new use, and many jurisdictions haven't yet set a precedent for tolerance and dosages in food-producing animals. This means that in many cases, applicants and regulators need to collaborate to figure out how to proceed when this uncertainty exists. Second, using evidence obtained for one intended use to substantiate a different intended use, even if the additive contains the same active ingredients, is inappropriate. For example, when different formulations, doses, or administration routes are needed. Let's examine differences and similarities next. The major difference is that each jurisdiction uses unique criteria to legally classify anti-methanogenic feed additives. The implications are the challenge to identify a single set of criteria for scientific evidence that satisfies all legal classifications, the varying perceptions of risk, the increased cost of compliance as targeted jurisdictions increase, and the existence of technical and economic barriers to trade across jurisdictions. The major similarities indicate consistency across all jurisdictions on the need for robust evidence for efficacy, safety, and product quality, and documentation, even if the type, size, duration, and location of the studies they require differ. Now we transition to the recommendations. You can read the three main recommendations for applicants on this slide. It is important to understand that in all jurisdictions, the burden of proof is on the applicant. In other words, the applicant is responsible for providing scientific evidence to substantiate safety and effectiveness for the intended use. For this reason, applicants and scientists helping them must understand and follow the jurisdiction-specific regulations and evidence requirements. For this reason, applicants and scientists are encouraged to consult with the authorities in each jurisdiction before designing and conducting studies intended for authorization on any intended use. The recommendations for scientists follow. It is important to understand that scientists conduct efficacy and risk assessments that are used by the authorities to make the risk management decision to approve or deny applications for each feed additive intended use. In other words, scientists quantify the desired effect and assess potential risks associated with each intended use, and authorities base their approval decisions on the scientific efficacy and risk assessments, along with economic, political, and cultural considerations to mitigate or eliminate the associated risks. Also, recommendations to follow good scientific practices is a good reminder for scientists and critical knowledge for applicants and the dairy sector stakeholders that use the resulting science to substantiate their decisions. As seen in these last three recommendations, scientists can also have active roles in improving regulations and communicating advantages, limitations, and appropriate use of feed additives to stakeholders to improve overall understanding and transparency on them. Thank you.
Thank you so much, Juan. Another example of a very complex part of the process and very nicely done and presented was something that is sometimes hard to understand for animal scientists, and it's very clear that we have an active role to play in different stages, even after the regulation is in place. So now we move on to the last one, which is about how we can account for the reductions in carbon emissions at different levels. As you know, there are now products are coming to the market, and now we face another challenge, which is put the tools in place to have an accurate accounting system. And this is going to be, well, this is led by Agustin del Prado and Roberto, and this is going to be presented by Agustin del Prado.
The following study is about accounting for the mitigation potential of anti-methanogenic feed additives. You can access the paper using this QR code. My name is Agustin del Prado, and I'm co-leading the work with Ronaldo Viuart. Here you can put a face to the whole team. Recent advances in our understanding of methanogenesis have led to the development of anti-methanogenic feed additives, AMFA, that can reduce enteric methane emissions. As such, one of the key questions for the full applicability is how to quantify their potential to reduce greenhouse gas emissions. In this study, we examine current and new approaches used for the accounting of enteric methane abatement by the use of AMFA from the individual animal to the global scale. We try to give recommendations on the accounting methods, and we will be talking about farm scale models, emission trading schemes, life cycle assessment, LCA, and regional and national inventories. The term accounting here mostly refers to the quantification of enteric methane abatement at different scales, and to a lesser extent to the quantification of uncertainties and potential trade-offs with AMFA used. Here we go very briefly on the main recommendations of the study. Fully accounting for the methane mitigation potential of AMFA is complex and involves several components that directly or indirectly interact with the AMFA. This includes considerations at the animal level, farm level, and broader system levels. Various factors such as site conditions, production types, and farm management practices play an important role. Additionally, we must consider boundaries beyond the farm, including the electricity or fuel used to manufacture the feed additive. All of these elements are interconnected and affect the overall potential for the mitigation. And first, an important recommendation: we need to tailor the methodology chosen for accounting with the right scale of the analysis, the availability of data, or even the objectives of your accounting. Perhaps an obvious recommendation, but these principles have not always been followed. We need always to differentiate between the efficacy of the feed additive. So we are talking about how well a feed additive lowers methane under ideal control conditions at lab scale or in trials with animals, and the effectiveness. The effectiveness is how to translate in real world this efficacy in reductions of methane emissions. The accounting also needs to consider AMFA delivery methods and the accounting of synergies and trade-offs in greenhouse gas emissions that can occur before the farm level. So for example, when the AMFA is produced and distributed, or even after. So what are the knock-on effects of this AMFA strategy on the feed efficiency of the animal? The assumptions on the additivity of AMFA abatement should also follow insights from experimental work, and in the future from models, and these continue to improve the simulation of combined AMFA efficacy and effectiveness. Full effect should be considered according to legislative and regulatory bodies. So some final conclusions for the different scales. So at the animal scale, relatively simple estimates of enteric methane emissions are often derived from prediction equations that include dry matter intake. There can be some more complex approaches that can be used, but they require much more information about the quality of the feed, for example, or may involve representing rumen fermentation and gastrointestinal digestion process, and this would be made by mechanistic modeling. At the farm level, it's essential to include sufficient detail regarding livestock and feeding conditions. The representativeness of these models also depends on their ability to accurately simulate a specific farm with a particular region. At the life cycle analysis LCA level, greenhouse gas emissions are evaluated across the entire production system from a life cycle perspective. So this is interesting. This assessment can help identify actually trade-offs in emissions at various stages of production beyond the farm boundaries. So for this, we need comprehensive accounting of farm emissions. In terms of regional and national inventories, to our knowledge, greenhouse gas emission inventories at these scales have yet to account for the use of ANFA. Any new mitigation measures introduced, for example, in the national inventory, it's good practice to provide solid evidence of the uptake of the technology, in this case, the ANFA technology in agricultural practice, and to apply it only to emissions from livestock where uptake can be actually validated. For ETS or emission trading schemes, recommendations will really depend on whether the point of obligation is set at the farmer or at the processor level. If it's at the farmer level, the accounting approach probably would be similar to that used for farm scale accounting. And in contrast, for processes, the accounting approach would likely resemble all the recommendations that are used in national inventory accounting. So with this, I come to the end. Thank you very much.
Thank you very much, Agustin and Ronaldo. I need to apologize, Ronaldo. You're not Roberto, you're Ronaldo. Yeah, I still find it hard to understand why someone from Argentina can be called Ronaldo and not Lionel. And in that duality, I got confused with your name. Now that we have the 6 papers presented, we have some questions already in the Q&A. You can add more if you wish. Some of them have been answered, but still can be part of the discussion. Now I pass the chair of the session to my colleague Andre Banning, who is going to be taking care of the discussion. All yours, Andre.
Thank you, David. So my name is Andre Banning, and I will try to guide us through the questions that have been posed. And I simply go through the first one, the question we see on top. Is the procedure of assessment a standardized method for publication or feed additive approval? The question was posed when Alex gave his paper, but I think it suits Juan quite well.
Thank you.
Doesn't it?
Thank you.
Juan, could you answer? Could you start answering?
Thank you so much. So definitely what Alex was recommending, well, are all recommendations on how to conduct proper studies to measure efficacy, safety, etc. At the end of the day, those same recommendations apply to regulatory processes and authorization procedures. So I would encourage everybody here that's on the attendee side to understand that these recommendations eventually will have, in terms of recommendations to conduct good science, will also have an impact on the regulatory process in every single one of the jurisdictions, because at the end of the day, the concept of a robust scientific evidence is a strong similarity among all of them. Hopefully that answered the question.
I hope so. And if it does not, they should raise another one in the questions and answers box. I go to the second question. Actually, one was already answered by Evert. I think it was a really interesting question, but I leave this one for now because it was already answered by Evert. I go to the question we have on top now. At what level of farm level activity data is needed? At the country level, to develop country and regional hybrid models to support greenhouse gas emissions estimations at the country level. So it's not that clear what the question is, but I think it's typically a question for Agustin. In what activity data, or Ronaldo, also good.
Good one.
Sorry. Yeah, I should have known.
Thank you for the question. Can you hear me all right?
Your paper, yeah.
Look, it's a good question, and it's quite a loaded one. I do have to confess. At a farm level, sorry, let's start with the animal level, minimum intake. As we know, intake is an already a good estimate, but in addition to that, we do need a few other things. But taking it to the farm level, it's probably knowing what and again, it makes quite a difference if it's a dairy or sheep and beef or some other category, because for dairying, and especially seasonal dairying, it's easy to follow the mobs and getting a little bit of mob intelligence and what's happening, animal numbers, parity, days in milk, etc., etc. When it comes to sheep and beef systems, there are many more growing mobs that you've got to account for. That in a very summarized response. At a country level, do you need thoughts? Heaps. I'll probably leave it at that and probably write something down for later on, because it might go on forever.
Okay, thank you. Thank you, Ronaldo. There's another question on the modeling side, so that one is for you, Ermius, to answer. Are you thinking about this just for methane reductions, or do you also incorporate other productivity consequences, feed efficiency, productivity of the animal, maybe even digestibility?
Yeah, so but primarily it is for methane emissions, but you can use a lot of the techniques for productivity as well, because in some cases there could be an impact on productivity, an impact on digestibility, and others. So if you have the data available, then those kind of modeling that we discussed on the paper could be applicable. For example, in the meta-analysis, you can look into what is the effect of the feed additive for methanogenesis, but also you can look at what is the effect on dry matter intake, what's the effect on milk production, or any other variables of interest as well. So yeah, I think primarily for methane, but other variables can be used as well.
Thank you, Ermius.
Andre, could I
Yes,
jump
of course,
in for just one second?
Other authors, join in, please.
Yeah, I want to make a quick comment about this question, because once again, the data depends on the intended use. So if an applicant were, for example, to want to have a claim about reduction of methane emissions intensity, they would need to provide data not only on the emissions, but also on the productivity. So that's why it's important in the regulatory system to understand that these recommendations are general, but they are tied specifically to the claim that the applicant is pursuing.
Thank you for this important addition, Juan. The next question is, will the synergistic effect between potentially anti-methanogenic compounds be considered? So are we looking at synergies between different anti-methanogenic compounds? Do we do that? I think for you, Evert, maybe this is an interesting question, or for Alex, maybe both.
I'm sure I can throw a sentence or two, but we have a whole section in our paper on this, and it's absolutely important because they could be synergistic, they could be antagonistic, and 1 + 1 may not equal 2. So there is a whole kind of combinations of factors and outcomes that can come from this. But I will encourage this listener to go to our paper and look at this particular section. There is, all the information is there.
But I think this is still a bit coming. This combination of additives is not that common yet in research.
Exactly, and we emphasize that we don't have enough data at this point and more research is needed.
Okay, thank you. Evert, do you have something to add on this?
Yeah, no, in the end, since the things that are available are not that potent yet, so you would think that in the end, probably synergistic effects going to be important. But that depends on what else will be found eventually. But
yeah, and Evert and Zoe, you're there as well. But I think it's not so common for your paper, the type of work that you described in your paper, to combine additives as well, is it?
No, but I'll jump into this conversation because in Australia we have commenced projects on this type of combinations. So we are combining some potent additives and different modes of action. So the work is underway. So yeah, watch out this space.
Okay. It's the near future. Thank you both. There's another question for you, Alex. What metrics for rumen fermentation and microbial profiles do you think are important to collect from animal trials? And how do these metrics compare to in vitro work?
Again, this is all detailed in the paper, but obviously the typical measurements that we take when we do animal studies for rumen metabolites, obviously volatile fatty acids. If we are interested in hydrogen, we are going to look at secondary or minor hydrogen sinks that again can be analyzed in the rumen. Other measurements are certainly important, but many times difficult to conduct. Microbial synthesis is one that we are missing in almost all of the studies with anti-methanogenic feed additives because it's difficult to conduct this kind of studies and the data are extremely variable, but that's another sink for hydrogen if you want, that we are missing all the time. In terms of comparing this to in vitro, I forgot, of course, the microbial data are critical. We are looking at microbial profiles using different techniques, DNA, RNA, and so on, and that's important, of course, as well. Between in vitro and in vivo, there is not a lot of differences in what we can measure, but obviously the results from an in vitro and in vivo will be totally different. And we have seen this over the years that very rarely in vitro outcomes from these additives compare one-to-one to in vivo. And that has to be kept in mind as well.
Yeah. It's always difficult but how, just another question I would like to put on top, how do we actually use these microbial profiles? Is it more to get an indication of the type of changes that are going on in the rumen, or can we also use it to quantify and to be able to get numbers on the extent something has been changing in the rumen? What do you expect?
My opinion is that this is a very, very fluid, if you want, field of data. We always analyze this, but in some cases, there are more to explain the effects or try to determine the mechanisms and the mode of action rather than looking at quantitative measurements. Yeah. So it's qualitative more than quantitative, I would say. And again, it helps us in many cases to explain what is going on with this treatment.
Yeah, thank you. Another question, what are the guidelines for evaluating the impact or the lack of impact of anti-methanogenic feed additives on productivity? Energy-corrected milk yield. So what guidelines, how do we, I think also for you, Alex, how do we actually determine these kind of things? What is necessary to prove, let's say, these impact on productivity?
Obviously, if we are strictly interested in gas emissions, methane measurement is sufficient, but this kind of additive that doesn't have production data will have no future, obviously, in animal nutritional production. That's why we need to measure more or less all productive variables. We need to measure daily gain, we need to measure milk yield, milk composition. We want to express also these emissions on some kind of a converted milk basis, whether it's fat and protein corrected milk or it's energy corrected milk, so we can account for potential differences in milk composition. We have to monitor body weight change, body condition score of these animals. In a long term, we have to monitor effects on health, on reproduction. So there is a lot of things that go into this equation, but at some point you want to look maybe at the major effects, and then further on down the road, those additional effects that require longer term studies or more intensive studies can come.
Yeah, I think that was the question also, directing at what guidelines. So what kind of studies do you need to actually demonstrate these effects on productivity? So we have these short-term trials, you have longer-term trials. So what do we actually need to really prove those kind of effects?
If I may, I also want to raise the point that every single one of those response variables that Alex mentioned will require a different amount of replicates to have the appropriate power, right? So, for example, determining a somewhat smaller change in milk production will require large numbers of cows that are not necessarily required to determine that a reduction in methane emissions when that reduction is large. So that's another thing to keep in mind, that the power will be different for the different response variables.
And I would add to this as well, Andre, that in many cases the power is not there to show or prove any effects. So in that case, it's a good strategy is to conduct more studies at different locations. Of course, if funding is available for whoever initiates this work, and then do a meta-analysis type of study that will combine these effects and maybe show a production effect or lack of production effect.
Yeah, the answer that Ermius gave just a moment ago, that in principle you can take those kind of animal responses also into the meta-analysis. Yeah. I'm sorry, Alex. This one question starts with Dr. Christoph, so it's for you as well, I guess. You have highlighted some techniques to underestimate methane emissions. I think to estimate is meant such as respiration chambers, green feeds, and SF6. You have already used the SF6 technique using 3-NOP and seaweeds in the diet. Have you even made any comparisons between the two?
Comparison between the two.
3-NOP comparing the seaweed. Yeah.
I don't think this question is very clear, completely clear anyway. But if we talk about techniques, these three techniques have been used, and when properly used, we recommend them to measure effects on methane emissions. How they compare with each other, again, this work has been done and published, not just by us, but including colleagues here on this call. So I would recommend that this listener goes and read the papers. I'm not going to comment on 3-NOP and seaweed and these kind of things, because this is not actually the objective of the paper.
No, and they both are potent in inhibiting methane, we know. So, but a really detailed comparison requires more time, and yeah. And I think the person asking this question, you can always reach out to the network, the Feed and Nutrition Network, or to members of it, or to presenters here, if you want to know more details about these measurement techniques and how they compare and how they can be used. The next question, how relevant does this area of research remain amidst public criticism and distrust, for example, as recently seen in the case of Bovaer? How does this impact on research funding? Yeah, I think who can answer this one?
I will take a shot at this.
Okay,
so
yes.
Because this has to do with public perception, and the reality of the matter is that, at least in the dairy sector, the the stakeholders will continue with their long-term goals of environmental stewardship and environmental responsibility, and therefore this doesn't go away because somebody posted their opinion, which by the way was very much disinformed, on Bovaer in the United Kingdom, and there was a flurry of in the media and in social media particularly, that eventually died out. So as far as I can tell, the dairy stakeholders that we work with continue to be committed to improving the environment, and therefore this will remain an important area of research.
Yes, that is also my feeling. Yeah, and it should also not be forgotten. But it's difficult to explain to the public. I think that there are regulatory bodies that really carefully check whether these compounds can be used. So the safety of their use, all these kind of things have to be delivered in dossiers. So there are committees that actually check what these additives are doing. It's difficult to get that. It's difficult to get that across to the public who make use of social media.
Yeah, sorry, and that's why I made the comment that the comments in social media were disinformed, because if a product like Bovaer is actually authorized for use, in this case it was in the United Kingdom, then all of those answers have already been supplied to the appropriate regulatory bodies, which is the case.
Yeah, but I agree with you that how does it impact the research funding? I think at the moment companies are still committed to contribute to this mitigation of enteric methane, and that, as far as I can understand, it keeps going despite of the unrest at social media. Maybe do other presenters have to comment something on this question, or should I proceed to the next one?
Can I just quickly, just to complement on what Juan said, I think for both researchers and stakeholders, I think it's very important to consider how relevant good communication is. So we can do good research, we can go through all the regulation processes, but if then the results and the impact is not well communicated, then we may go into situations that are not very pleasant, and that's the example of Bovaer. So this is something the animal scientists sometimes we struggle with, because we are not it's not on a daily basis activity, the good communication with different stakeholders. But this is something we need to be very, very careful on using the right wording and the right message at the right time to the right audience.
Yeah, I agree. Yeah, and the basic mistrust that sometimes exists against scientists, that's almost unavoidable. There's not much you can do against that. Yeah. Let's see. The next question is from Ferreira. An effective accounting method is accompanied by a strong MRV framework. Personally, I'm not sure whether I understand the abbreviation MRV. Any recommendations regarding feasible MRV? Sorry?
Andre, MRV stands for measurement, reported, and verification.
Okay.
Yeah.
So now Ronaldo also knows what it means.
Yeah, you should go, Ronaldo, because it's
I think so. It's for Ronaldo. Yeah. Any recommendations regarding feasible MRV for methane-reducing feed additives?
I do apologize, because I was going to take it completely different. Thank you, Juan, for intervening. Of course, the measuring and verification are absolutely critical for this. And the more data that surrounds these verifications, the more effective we can come up with a recommendation or a conclusion. My mindset, for some reason, went off around scanning and imaging as a support tool in that kind of framework. And look, I wouldn't be surprised if we're going to see more and more of that, particularly around scanning. But anyway, I won't go into any detail. It has nothing to do with that. It has to do with what Juan said. And ideally, yes, it really depends on the data supplied and being able to come up with a recommendation after these verification techniques.
But it indicates a whole framework, but I wonder whether the framework can be the same for every type of additive. Maybe it's not. You can have a framework, but I think the details that matter will also depend on the additive you're actually looking at and the way it's being applied to animals.
Absolutely.
But our recommendations in how to do this in all of the 6 papers, in how to study them, but also how to account for their efficacy.
I think particularly for this question, I would encourage that whoever's interested in putting together an MRV process use Ronaldo's paper and Hermia's paper, the ones that are on modeling, because frankly, in most cases, those are the types of modeling approaches are going to get used for putting together an MRV system. So I would just recommend that you follow those recommendations in both of those papers.
Thank you for that addition, Juan. If there are no further additions to this question, I go to the next one. Is there a decrease in feed intake without a decrease in productivity? Is this seen as acceptable? So maybe Alex?
Probably talk a little bit about this. That's actually a very good question, an important one, because you do see these kind of effects where feed intake decreases, milk production eventually catches up. At the beginning drops when feed intake drops, but then eventually climbs up to the level of the control, for example. When we are looking at this, though, we have to be careful and look at the whole picture. And the whole picture includes, as I said before, body weight changes and body condition score changes of these animals, because they may still be maintaining milk production if they are milking animals, but the energy may be coming from their back. So it's important that we have these long-term studies where we can collect data for these variables and put it in an equation on feed efficiency. Feed efficiency is one thing, but then feed efficiency, including body weight loss and body condition loss, is another one.
Yeah. And small changes that you hardly can distinguish in body weight changes, they may have a big impact on the productivity that you see. It's a lot of energy in that body weight, so it easily affects productivity or matches for the lower feed intake. Yeah.
No, I wanted to make this clear, but of course it's possible that feed additives improve feed efficiency without affecting, weight loss of the animal or gain. These are rare situations, and the changes will be small, but it's possible. It's not impossible.
Yeah, we also had it in one of our trials. Yeah. The next question then, unless somebody wants to jump in from the presenters. How can this community as a whole move forward on these recommendations most effectively? It is not duplicating their efforts. What groups and efforts are being set up here? Well, you are the chair of the Feed and Nutrition Network, David. Maybe this is a good one for you to answer.
I think this is a very important question. Well, first, putting these guidelines in place was not easy, so this is a very good first step. But I agree that what comes next. What we're doing now is the second phase, which is trying to disseminate this as much as possible, not just only publishing in Journal of Dairy Science, but also organizing these webinars to discuss with the scientific community, but also to participate in other type of events with stakeholders, with the private sector. And Andres also attending a meeting of the Global Dairy Platform soon, also to discuss the guidelines. So trying to disseminate this as much as possible. And then since this came from the Global Research Alliance, and in particular the Feed and Nutrition Network, there is an active group that's going to keep working on this. Obviously, we don't have control, or we are not an international evaluation panel for anything, but of course we can remain as also a group of experts to consult in case there are further questions or further requests from anybody. But yeah, the Feed and Nutrition Network for sure is gonna. It's been very active so far, but it's gonna keep being active in this regard. And there will be also other events at the Animal Dairy Science Association meeting ATSA in June, also at the EAAP in September, and at the GGAA meeting in Kenya in October. So this dissemination phase will remain for a few months to make sure that this reaches as many people as possible.
Thank you, David. I think we can do a last question before we have to wrap up this webinar, and we are close to 1:30 over here. The next question is, we have some evidence that the prebiotic we have developed increases the rate of clearance of fermentation products, ostensibly reducing the availability of substrate for methanogenesis. How is interaction with other rumen processes considered?
Yeah, I can take that one if you want.
Yes, Alejandro. Hey, there you are. I
Yeah.
see you for the first time now. Sorry.
Yes, basically in our paper we have a section which is dedicated to this particular thing, which is how to shift the fermentation pattern in order to lower the hydrogen production. I will not call it prebiotic because at the moment we don't know any particular prebiotic which is acting as in the question is described. But there are a number of feed additives which are decreasing the hydrogen production. For example, the tannins can act as a can generate a binding with the proteins and the substrate and decreasing the fermentation of the diet. So when we normally conduct in vitro trial, it's important as well to check the total volatile fatty acid production, because when we incorporate the additive, in many cases we observe a decrease in the fermentation. So it's very important not only to keep the fermentation going on, but as well trying to decrease the hydrogen. So yeah, I think it's relevant to decrease the hydrogen production.
And there's another one for you immediately. I think it's the mode of action guidelines paper, and it asks on what topics do you place higher importance than others? You mentioned many details, and it's complex, that's obvious. But some areas of research may be you may consider more important than others. Which one do you consider the most important one?
Yeah, that's a good question. The first thing we would like to say is that understanding the mode of action is very complex. So these studies are only recommended for those additives which have been already proved that they are effective and they don't have any problems with the safety. And the key areas of research for understanding the mode of action, we will say that are understanding what is the effect on the rumen fermentation, if it's shifting the fermentation towards a lower hydrogen production, if it's inhibiting specifically to the methanogens, that's another type, and within that there is a broad number of methodologies. And then another area of research which is at the moment relevant is trying to find alternative electron acceptors, because at the moment we have found with the 3-NOP and with the bromophone additives which are able to decrease methanogenesis, but there is still an excess of hydrogen that is not providing any benefit for the production. So if we manage to find an alternative electron acceptor, we could redirect the hydrogen towards some volatile fatty acids, other products which could have a win-win scenario.
Okay, yeah, Yeah, okay. So that's to come for the future then maybe, yeah. I think there's another interesting question which is on in vitro. So Evert or Zoe, or you both, in testing in vivo anti-methane effects of the, oh no, it's on in vivo, sorry, but it's, is it necessary to balance nutrients, isonitrogenous and isocaloric? Yeah, so it's for Alex, in principle in vivo, but I suppose for in vitro it holds just as well. If you are testing additives, you also need to think about what nutrients are you supplying in your batch culture or whatever. Yeah, but Alex, can you answer this one?
Yeah, if I understand it correctly, of course, when you have a treatment diet versus a control diet, and the only difference is the feed additive, then the rest of the diet has to be balanced and they have to be the same, so you don't have confounding factors of any kind. If your treatment is not a feed additive, but changes in nutrients and feeds and fermentation patterns and so on, then the diets will be different. But for feed additives, certainly the basal diet has to be exactly the same.
Yeah, you have a control and you have the same diet testing the effect of your additive. That's what you want to compare. That's the aim. Yeah. Yeah. So other questions, they have been answered already, I see. What is... Yeah, then a question, what is important? The absolute emissions or the emission intensity?
I have a comment about that. Once again, it relates to
Juan,
relates to the intended use. So an applicant could, for example, put together research along with the scientists to have an intended use that basically says that the purpose is to reduce emissions intensity. Okay. Now I am going to say that from the supply chain perspective, intensity is super important because the companies that purchase milk want to purchase milk that per kilo of milk has less emissions, right? Now, obviously, for the world as a whole, absolute emissions are important because at the end of the day, is the total amount of greenhouse gases in the atmosphere that have the greenhouse effect. So I don't think this is a question of either or. I would never post it as what is important, absolute emissions or emissions intensity, because both of them are important. So I would say these are both important. I will always use the and explain why the two of them are important.
Yeah, you want the whole picture. Yeah. And there's another paper which is Zoe actually answering at the moment. She is typing now. So I go to the last question then. That's in the whole list of questions. How do you incorporate the outputs of an LCA for a feed additive into a farm level or country level greenhouse gas inventory? So the emissions from the production of the additive is included alongside the emission reductions from its use. Well, this must be a question for you, Ronaldo.
Oh, thank you. A little bit of a hospital pass there. We do provide some insights in the paper, so please read the paper. But also, I'd like to say that with every kind of emissions reduction or emissions abatement, be it a feed additive or whatever it is, you do really have to factor the effects beforehand, before even testing it. What does it mean in terms of production, in terms of greenhouse gas emissions? These things have their own profile. So you do have to enter that into account. Now, when or how is that going to be accounted at a national level? That's an interesting one, because it will have to bring a lot of not only mind power, but an agreement that is the case. I don't think we've done a good job on it, but LCA is a growing beast. And I think incrementally, these things will be accounted for. For feed additives, they usually are. People have done quite a good job, but it's not applicable to all enteric methane abatement technologies or techniques out there. So look, watch the space. I think it's absolutely critical to account for the whole stage, for the whole life cycle.
Can I jump again? Sorry, I'm very
Yes, you
talkative
can.
today,
You
but
can. Yes.
I want to say that Ronaldo's answer is wonderful, and it reflects what the reality is. Here in the United States, we have this challenge. One of the issues is the incompatibilities between the different methodologies. The inventory methodology may not be able to capture the effects of the feed additive. We see that here in the United States, for an example. The methodology is just basically blind to the feed additive effect. Another challenge is that in many instances, if the methodology were able to capture that information to incorporate into the inventory, you'd still need activity data. You would still need to know how many animals in your country are consuming that particular feed additive. And that data is not easy to get to.
Just going to add, Andre, very quickly that we've made an attempt here in New Zealand around nitrification inhibitors. Oh, I think we're gone. We might be gone.
Yeah.
We hear you. We hear you.
We hear you.
Brilliant. I did have a little bit of a meltdown. Sorry. There was once upon a time with nitrification inhibitors, and it was a very coarse accounting. It was just around consumption and assuming best practice for application. But I think we need to be better than that. And of course, we're going to be challenged with accounting for the whole cycle. Thank you.
Thank you, Ronaldo, for this answer. I think we had this one already. Yeah, that was just answered. There's another one. What's the carbon footprint of feedback additives as a product? I think these companies, maybe I can answer this one. These companies, they should do the LCA, figuring out what the greenhouse gas emissions associated with the production of the additive are. So those kind of numbers they should deliver if they want to end up in any accounting system or in a national inventory. I think that belongs to producing those kind of methane mitigating feed additives. And I think companies are doing that actually. We have the whole list of questions more or less answered. There's still a few, but I think I give the word back to you, David. Because it's getting later and later, and it has been quite long already.
Yeah, it's been 1 hour and 40 minutes so far. I think it's been quite an active and interesting discussion. I'd like to thank the group leaders who prepare the presentations. We know that it's really hard to put into 7 minutes or 6 minutes a long paper and a lot of work done over 2 years, but it has really facilitated to leave space and time for the discussion. I'd like to thank all the participants and for waiting and then sending the questions. So we're still answering some of them. We will check through the questions and again, if when we finish the second webinar, we will post and send the questions and answers to everyone. So we make sure that you all have that piece of information. There might be some different questions coming in the second webinar that you might find also interesting. And well, I'd like to thank Chista and the GRA for the support in preparing the Zoom and the logistics to have this session, and of course to Florencia Garcia, who probably, if you can turn your camera on, Flor, maybe we can see you because you're always behind the scenes, but it would be nice if people can see your face as well. Flor has been supporting us in the whole process in many aspects, not only scientific, others in organizing this work over the last 2 years. It is now two years or more. So yeah, quite hard work that Andrea and I could have not done without her help. And one of them is of course preparing these 2 webinars. So thank you, Flor, for your support. And yeah, with that said, I think we can either go to bed, those that are in in the middle of the night, or go back to your office or whatever you want to do. Thank you again, and well, some of you we will see each other in 2 days, I think. All the best.
If anybody wants to reach out to us about the webinars and want to know more about the whole project or the work that has been produced in this project, you can reach out to me or to David Yanes, and you see the emails on the screen now. So you can simply send emails for further information. And with this, I want to give the word to David Yanes, because David is hosting the discussion section. We have several questions I saw. David, the word is yours.
Thank you, Andre, and thank you to all presenters, and good afternoon or good morning, depending where you are. Thank you for the questions. I think we had quite a nice list of challenging questions, I would say, and I will try to address all of them and direct the questions to some of our colleagues, but for the working group leaders, of course, if you feel like you can also provide useful feedback, please turn your camera on. So the first one, but we have two questions from Frederick. Hello, Frederick. Thank you very much. One is related to how hydrogen can be detrimental in terms of rumen fermentation when it accumulates as a result of the application of an anti-methanogenic feed additive. And I think we can provide probably an answer on the metabolic and then maybe more the rumen function way side. So maybe we can start with Emilio, who can, to me, is the world guru of hydrogen flows in the rumen. So maybe, Emilio, if you can provide a short as possible explanation of why hydrogen accumulation may be detrimental, and then we can move on to more the practical side from those like that done some animal trials like Peter Land on the second paper.
I'll address the problem of hydrogen accumulation from 3 different aspects or point of view. The first or 2, I'll go through them quite shortly. In terms of by loss of energy for the animal, it is moderate compared to the energy that we saved in methane that's not made. A second aspect to consider is that hydrogen has some consequences on heat trapped in the atmosphere. It is not a greenhouse gas per se, but indirectly it does cause some heat trapping. But that has been estimated to be less than 1% of the heat trapping capacity of methane emission of methane that's suppressed. Again, that's a nice recent paper by Alex Christoph. And then I'll talk about rumen fermentation. Historically, there is a concern that hydrogen accumulation could inhibit NADH deoxidation and hinder glycolysis and fermentation. In vitro, the meta-analysis that's been shown to happen, there is an inhibition of fermented hexoses. But in vivo, the empirical evidence is that when we inhibit methane production and the hydrogen accumulates, the digestibility, at least apparent digestibility and fiber digestibility, is not compromised. How could that happen? There is a shift in hydrogenases from conjugative hydrogenases that reoxidize NADH with ferredoxins to fermentative hydrogenases, and the NADH reducing equivalent gets redirected to other metabolic pathways, propionate and butyrate and ethanol as well. So that's what we know about this the consequences of hydrogen accumulation and fermentation.
Thank you, Emilio. And I know it's a complex issue, and you wrote some articles about it, so maybe people can go and read those articles. Maybe Peter, I don't know if you want to comment on the practical experience that you have had with different antimethanogenic additives and how hydrogen the increasing hydrogen production can be detrimental overall, and maybe link it to dry matter intake if that's relevant.
Yeah, I fully agree. I mean, we had some years ago, we had this hypothesis that increase in hydrogen was also associated with a reduction in feed intake. But if we look a little bit closer and look into what actually happens in the rumen, and just alongside what Emilio just said, when we use some really potent feed additives that reduces methane significantly, when we then measure the digestibility of, for example, NDF, so rumen digestibility of NDF in cannulated cows or total tract digestibility of NDF, we don't see a negative effect on the digestibility. If we see an effect, it actually seems to be positive. And the other thing is that we have done some trials where we use the different dosages of feed additives. And we could see when we had the high dose in play, we saw some reductions in feed intake, which were not as pronounced and not even significant at the low doses. But the hydrogen emission was exactly the same. So whether we used the high or the low doses of the feed additive, emission of hydrogen was the same. So that showed that hydrogen was not linked to this reduction in feed intake. So my point of view for now is that I don't think that this hydrogen is directly linked to reduction in feed intake or reduction in digestibility.
Great, thank you, Peter. I'm sure we can continue this conversation about hydrogen for longer, but we've got other questions. The second question from Frederick is about the impact on the methane produced from manure, if there is any indirect impact of feed additives on methane production from manure, if the objective is to increase or produce biogas. I don't know if you guys in Wageningen, Andre, or Peter in Aarhus University in Denmark, you have experience on animal trials and the indirect effect on manure or methane production from manure.
Can I start, Peter?
Yeah, sure.
If we test our feed additives, and in those trials that we did check digestibility, most often we don't actually. Depends a bit on what kind of trials you're doing. In respiration chambers, of course, you know digestibility, but if it's a practical feeding trial, it's not so common. But in those trials that we did measure digestibility, I have the same opinion as Peter had. We don't see that much change in digestibility. So I would say adding an AMFA to a diet, and if the diet digestibility doesn't change, also the biogas potential stays the same. It's the same nutrients that end up into fecal matter, into urine, and that delivers you the manure. So I don't see that much changes. Of course, it depends on your feed additive. If it's something like fats or fatty acids, then digestibility may change. If it's something like nitrate, then you add a lot of nitrogen in the diet, and you may end up with a higher nitrogen excretion by the animal, and then you have a trade-off towards more nitrogen. But biogas potential, I don't think it will change that much. That's my impression.
Yeah, and we found exactly the same when we have collected manure from animals fed different feed additives and tested for the biogas potential in vitro, so in small bottles. We don't see any change. We might see an effect of when we add fat to the diet, but the most important thing that actually, you can say that affects the biogas is how you store your manure afterwards. So whether you store it outdoors or in a high temperature or low temperature, that was in our case the main driver for biogas potential of manure. It was not the feed additives or the diet composition.
Thank you, Peter, and thank you, Andre. I think Emilio turned on his camera, so please go ahead.
Just to say, there is a couple of papers that show that animals, the manure from animals that were fed CNOP was not affected in terms of methane production. And also the case of bromoform from Asparagopsis, I don't know of such an experiment, but bromoform was not detected in feces, nor was dibromomethane, which is the reduction metabolite of bromoform in the rumen. The reason in both cases has to do with the short life of both active principles in active compounds in the rumen. They are metabolized quite quickly, so they don't get to the manure.
Yeah, that was a great point. Thank you, Emilio. Now we'll move on to the next question, which is how can we check and ensure if reference sources used are peer-reviewed? I'm not sure if the question relates to the references used in these 6 papers. If that's the case, all the papers have gone through peer review process in Journal Dairy Science. But I don't know if the question relates actually to the registration process with the regulatory body. And in that case, I know better the EFSA regulation in Europe that needs to go, the dossier needs to go through a scientific panel evaluation. So scientists are part of the panel, evaluate the information provided and the references. And I don't know, Juan, if you want to add anything in relation to the evidence and the references provided during the registration process.
Yeah, I mean, at the end of the day, any references usually have to be peer reviewed, except that in some registration processes, there are opportunities to provide information that is not peer reviewed, is not publicly available. Usually, that type of information that it's not peer reviewed can only be provided for the purpose of validation, but not for the purpose of creating the claim. So if that makes sense, a company will provide a body of work that is used to create the claim for that specific intended use. And then they can use some information that is proprietary for the validation purpose. But once again, not for creating the claim.
Thank you. I think that was a very good clarification, Juan. Right, next question. It's very relevant. There is some discussion about the long-term efficacy and how we can make sure that the effect of any additive lasts for long enough. And the question is about what is the ideal time period after which the effectiveness of a feed additive should be reevaluated due to possible microbial adaptation, 5 years or so. And I know there are discussions about this issue in different international fora. I would like to bring to this question first Andre and maybe Juan and whoever wants to add as well. Andre, I think the longest trial published using 3-NOP so far, I might be wrong, comes from your group. It was a 1-year trial, I think covering maybe part of 2 lactations. So yeah, if you can provide information on what you found and the challenges to really prove what's happening after a long-term exposition to the additive.
Yeah, I think it's really a relevant point because we need to know, of course, if you account for some effect, we need to know whether there's adaptation or not. So indeed, David, we tested it for 1 year for 3-NOP in lactating cows, and it included the dry period and the calving period and starting up of animals again. The challenge is that it's highly dynamic. Diets are changing, silages that you're feeding are changing, the amount of concentrate that you feed is changing. So the challenge is to do it in a very balanced manner, that you can always compare the control treatment with the 3-NOP treatment. We did that in a trial and we saw huge variability depending on the diets and the changes of the animals. Also changes in efficacy of 3-NOP, and going down and up. And our impression is it was really dependent on the quality and the type of the diet. So at some moments the efficacy went down, but at the end of the whole 1 year period, it went up again. So that's counterintuitive. If something has been adapting, then you don't expect that the efficacy goes up again. And when we checked it with data on the quality of the diet, that gave us the most clear indication of good correlation between quality of our grass silage and how effective the 3-NOP was. And I expect this actually myself for other additives just as well. For example, for bromoform, I would expect similar things. For additives like nitrate or fats, I think it's different. I expect there are less of these adaptation effects occurring. For the inhibitors, I don't expect it at all. My main worry with adaptation would be on the class of additives that's plant extracts, all kinds of secondary metabolites from plants. And I think for decades already, these adaptation effects have been described already for plant extracts. So I think there most focus is needed on this point.
Thank you, Andre.
From the perspective of regulatory frameworks, the answer to this question is very easy. What is authorized is an intended use. That intended use not only it means the purpose, but it also has all of the how do you use it, all of the elements related to how. So actually the amount of time is part of that how. So if the evidence that a company has basically says that, basically shows, supports that the mitigation occurred over a period of 90 days, they won't be able to claim mitigation over 90 days. And so I suggest that everybody that has this question, pick up a label of Bovaer, for example, and read the fine print. And if you do, you're going to find that it says data supporting effectiveness has been evaluated when fed for no more than 105 days. So right there on the label of the product, there should be a statement that talks about what the evidence supports in terms of feeding period as well.
Yeah, I agree. But one thing I want to add, Juan, is that at the same time we have farm assessment systems, and they want to address the effect of something like Bovaer or other additives on the long term. So they include other kind of evidence different from the registration process. So they already put it into farm accounting systems, even though the label says it's only for this 104 days.
Yeah, clearly we have a challenge there for the accounting systems and the inventory systems on national level when they start being implemented. But yeah, it's a very good point from Juan. The claim and the it's very important, and the intended use, because that's what has been scientifically evaluated.
So this question says for GHG compensation projects. GHG compensation projects, the way that I interpret it, is related to some type of carbon credit or insetting. Usually those projects per se have a limited time as well. Usually those could be an annual project. And so in addition to whatever the label says, those projects already have some kind of a time frame attached to them. And that's going to depend on which organization is actually creating those credits or those inset values, and then how that verification process goes. So there are some also corporate policies that are attached to the processes that they use that also need to be taken into consideration.
Yeah, thank you, Juan. Right, let's move on to the next question, which is linked to the topic. So maybe Andre, you stay here if you don't mind. It's a question from Verley. Thank you for Verley on the variability of the effect of some additives depending on the basal diet. And you mentioned indirectly about the plant extracts, for instance. So to what extent could be a limited number of in vivo trials be complemented by modeling in order to limit the number of experiments? I know this is quite complex, and you've been involved in some meta-analysis with Jan and Ermias on 3-NOP. But I presume Verle's question as well is related to other types of anti-methanogenic feed additives.
Yeah, it's a very good question. It's also a difficult one to give a simple answer to, because this variability is quite complex and difficult to grasp. But indeed, I agree with Vele that if we look at feed additives, the number and types of diets and production conditions we have, we cannot test all the conditions and all the diets. And then thinking even about combinations of additives, it gets even worse. It's unaffordable. It's too much. So it would be really a good way forward if we are able to model those effects and the variability of those effects. If that would be possible, that would help a lot, I think. And meta-analysis, as Ermias presented in his presentation, that's one way of doing it in a more generic manner, putting all literature together and get some generic prediction equations. If you really want to do it based on an understanding of why that variability is coming forward, then you have to dive a bit more into the deep, and that part is a bit more complex. But I personally believe it can be done. It can be done. And plant extracts, that's the most difficult one again, because then you need to know what is the mode of action and what is actually happening in a rumen. As soon as you understand what is happening and why methane is going down, then it should also be possible to model it some way.
Thank you, Andre. Right, let's move on to another question by Sinead Waters from Ireland. Thank you, Sinead. Oh, sorry, Juan, you want to add something?
No, I'm going to answer the question that Sinead asked.
Oh, go. God, you're fast.
So she's asking about changing the format of a feed additive to a bolus for grazing systems. Have to go through a similar regulatory process, or has that been considered? And the answer again is straightforward, in the sense that even if the same ingredient is being used, if the form of administration changes, there needs to be additional supporting evidence to demonstrate that that new form of administration is safe and effective. So that's a different intended use, and authorization is based on intended use. So the answer is the straight up answer is yes. Additional supporting evidence needs to be provided to authorize that specific intended use through the bolus.
Thank you, Juan. That was very clear. And a very important question, because there is a lack of studies on grazing systems using different approaches, either bolus or drinking water, etc. Right, next question is by Elsa, and maybe Agustin, you can address this question. How should we address integrated uncertainty when using empirical models in practical farm national level applications? There you go.
Okay, thank you. All right. Well, it's one of the questions actually that was in the subsections of the paper. So there's quite a lot of detail, and we are quite lucky that the person that mainly developed that is here is Andre. But I mean, I can answer and see if Andre can complement my answer, and if it's enough what I answer. Well, I mean, I think the point is it's this challenge about incorporating uncertainties is not just for having like an accounting of the effect of ANFA. It's already for the basic accounting on greenhouse gas emissions for both an inventory or a farm level model. There is a challenge on how to account for uncertainties from many levels and from many sources that can happen. This not being sure of the data that we have is actually covering exactly what we are simulating. So, for example, I mean, for the inventories, actually, the recommendations are easy, because it's just try to follow the guidelines from the IPCC of how to incorporate the uncertainties. And obviously, if we don't have a statistical information about how this ANFA is affecting on the reduction of methane, it's going to be very hard to incorporate this uncertainty. So that's why actually I think Andre in some of his sections, he already mentioned that. I mean, it can be complemented by some, because we are talking about empirical modeling, it can be complemented, the empirical modeling simulation with the mechanistic modeling simulation. Once we can incorporate more data from trials, the uncertainties will be actually being shortened, will be smaller. At the farm level as well, well, again, you need to have statistical information. For example, if we think about the farm models in terms of just specifically on parameters, that's important parameters for the methane quantification, like the YM, the methane conversion factor. So how much of the energy of the animal is actually going to methane as a loss. For example, as a rule, I think the national inventories, the IPC was giving, like, for example, for dairy cattle, it was given in 2006 like a 6.5 as an average, and then 1% more or less as an uncertainty. But more complex modeling would have tried to do some more sophisticated methods like Monte Carlo, which implies, again, having knowledge on the statistical, the probabilistic distribution of how these parameters that are used for the modeling are propagated. So if you don't have that information, it's you can hardly use it. So I don't know if this covers a little bit the main idea, Andres. Sorry.
Yeah, I think so. I think it's really good, Agustin, and I agree with you. The only thing I would like to add is that at the practical farm level, there are all kinds of different systems that are in place in different countries. Things are represented differently. And with the more detailed modeling, we tend to focus on at the animal level. But maybe an animal is defined differently in your farm assessment system. That could be. And every system has a different approach. So what is really needed to answer this question is, for answering this, I think you really need a good documentation of how you handle this uncertainty. You need to put it down into a specific study focused on this aspect. It needs to be documented. How you handle it, what did you assume, how did you generalize, how does it affect the outcomes if I simplify the dosage that went into animals by just averaging it over a whole period. Those kind of details need to be documented. I think that's what's needed in addition if you want to introduce it into a practical farm assessment tool.
Okay, thank you, Andrea and Agustin. I will call Juan before he turns the camera on for the next question. Juan, this is I think it's a very relevant question about the patenting some products claiming methane reduction and only being tested in vitro, and how this could be regulated. I'm sure you have a clear answer to that, Juan.
Yes. So once again, the answer is fairly clear, and it's actually fairly short. The regulatory authorities in your country are the ones that regulate. Nobody else can regulate. So we did not examine Mexico in this particular study, but we examined 7 other illustrative jurisdictions. So I couldn't give you specifics on who to call in Mexico, but I wouldn't be surprised if the situation was somewhat similar, because the similarity is that everybody asks for robust evidence. So I want to make a couple of comments here on how the elements that were worded here in this question. First of all, patenting is one thing that has nothing to do with authorization. Okay, patenting is a process by which somebody is trying to protect their intellectual property. And so if somebody comes and tells you, hey, my product is patented, that doesn't mean that they can actually market that product for that specific purpose. The only way that they can market a product for a specific purpose is to obtain authorization from the relevant authorities in the jurisdiction or the country in which it's going to be used, right? And if there are products that are being marketed or are being discussed as claiming methane reductions in your country, and the authorization to market those products in that way is not available, then unfortunately, those individuals are doing something that they shouldn't be doing. And again, it's the regulatory authorities in your country who should be contacting them and telling them to stop until they provide the evidence that's required to obtain the authorization. I hope that that was clear.
That was very clear indeed, Juan. Right, next question is about what countries, from Laura Cardenas. Hello, Laura. And I think maybe Juan as well, but Peter, coming from Denmark, which I think is one of the countries that maybe started some initiatives at national level in advance of other countries. I'm sure that we can provide a list of countries that are already in the process or including already feed additives. I know that in Spain, for instance, Bovaer is being used, but maybe Peter, you can give a more detailed answer in terms of national schemes, and also Juan after.
I just want to point out that Laura also added a comment at the bottom that says that her question was incomplete, and it says what countries have implemented additives in their inventories. Okay. So I just wanted to raise that.
Yeah, no, thank you, Juan. Yeah. Then Peter, I'm sure you can provide some insight on that from Denmark.
Yeah, I can say what we have done in Denmark, and that was that of 1st of January this year, all the dairy farmers, or the farmers with more than 50 cows, which are almost all the dairy farmers in Denmark, and not the organic farmers, the conventional farmers, they were given an option. They should either increase the fat level in the diet over the entire year, or they should use Bovaer for 80 days. And I think it was around 90% of the farmers actually chose to go with Bovaer. And yes, this around 10% with increased fat level in the diet. And it's mandatory. You have to choose one of them. And it's compensated by the government. And actually, if you go for the Bovaer solution, it's actually also partly compensated for by the big dairies. I can honestly say it is something that has caused a little bit of disturbance in the media, because this is something new. I usually say it's the biggest difference or the biggest change in how we feed dairy cows that has happened in Denmark in the last 30 years. But I think there's also an understanding of that things are necessary. We need to do something. So I think that's kind of the status from Denmark. And the next thing is that it has to be, of course, because there's so much political interest in this, it has to be included in the national inventory. So what we have done is to say that, yes, if you feed Bovaer to your cows in Denmark, we expect that you can reduce your methane production with 27%. We know that that covers a huge variation between farmers, but that was to get started. So everybody gets the same reduction, 27%. And then we hopefully in the future, we can differentiate if farmers feeding maize silage, if we can document a higher reduction potential in those feeding situations, well, then of course, these farmers should have a benefit from that. But for now, we work with one reduction, a fixed reduction potential.
Thank you, Peter. Okay, Juan, and then Andrea and Alejandro.
Yeah, very quickly, in the United States, as far as I know, as of today, the only product that is authorized for reduction of methane gas emissions per pound of dry matter intake in lactating dairy cows is Bovaer. Again, a good example of how an intended use needs to go through an authorization process and the type of information that needs to be presented on the label. I don't know if any other product has that authorization as of today. I don't believe so. Second, the inventory, as far as I know, from the draft inventory that is on the EPA website, again today, cannot handle this. So there is no way in which the inventory can account. I am actually curious myself to find out what EPA is going to use in order to incorporate this, because products will get used here in the US as well, and they're going to have to they're going to have to take a stance. The final comment is that in order to make any type of calculation, the inventory will have to then get some level of activity data. That means figure out how many farms are feeding these additives, and that's not easy information to get to. Thank you.
Thank you, Juan. Andre?
For our case in the Netherlands, it's a bit different. Fats and nitrate and Bovaer, they are all being accounted for in our farm assessment tool, and which is also the tool that's used by the big dairy companies. And farmers get compensated for the reduction in methane. Not fully, not all costs are compensated, but it's on a voluntary basis. And yeah, simply the equations from the meta-analysis that David mentioned earlier, those kind of equations we use in our farm assessment tools. And the government is just waiting for farmers to really pick up additives before they will change the inventory methodology. And it's about now that we start our discussions. Okay, farmers actually start using it. How do we apply this or implement this in our inventory? We are not that far yet. That discussion is just coming now.
Yeah, similar situation. Thank you, Andre. We have in Spain, Bovaer has been used for more than a year now, and it's not been accounted in the inventories yet. And we hope that the papers that we published can contribute to that. But I think we mentioned this and we discussed the measuring, reporting, and verification being very important in these processes, and it's something that is still coming. Right, I will take the next question very quickly, unless then, or maybe Alejandro, do you want to take? Alejandro, you take the question.
Okay. Yeah, thank you.
Well, the question, sorry, is about if batch cultures in vitro systems can, well, provide some information about the kinetics of methane production in comparison with the continuous culture systems like or semi-continuous like Rusitech, and how close they are to accurate estimations of in vivo.
Yeah, understanding the kinetics of methane production is relevant because there is a diurnal variation in the methane production, as most of the methane is produced after feeding. So that is relatively difficult to mimic in vitro. But I will say that both methods, the batch culture and the Rusitech, they are complementary. For example, the batch culture and the new system, which is the ANCOM, allows to measure a lot of treatments with different diets, with different dosage, and we can measure, and as well as quick, it's cheap, and it's very simple. The problem is that it doesn't simulate the diurnal variation, as normally is maintained only during 24 hours. And as well, there is not time enough for the microbial population to be adapted. But okay, we can have a lot of measurements, but they are not accurate enough for describing the kinetics. The Rusitech is a step closer to the in vivo because we can keep it running for 2 or 3 weeks, and so the microbial population can be adapted. And as well, we feed the Rusitech once per day, so we mimic, or twice if we want, so we can mimic the diurnal variation, which can be relevant for those additives which they have a very quick mode of action, so they inhibit very quickly. For example, the 3-NOP. So yeah, I would say that they are complementary. And in relation to how close is to translate those results to in vivo, that's a gap of knowledge because particularly in terms of the dosage, we always struggle when we have to translate the dosage. But yeah, I think the Rusitech dosage is closer, although it's a quite diluted media, the one we use.
Thank you, Alejandro. I think that's very clear on that, how they can make an indication, but not really be so close to the rumen, which eventually is the real ecosystem. There's a question from Eva Ramos from Spain on if we can provide insights on the evidence or not of the effect of productivity benefits for farmers. I think, well, Emilio, it's already on. If you want to comment on that, or was it from a different question, Emilio?
It is that one. I published some meta-analysis in 2018 in which I I didn't find any relationship between the efficiency of milk production and growth with the inhibition of methane production. But that meta-analysis is a bit quite outdated. That was in 2018. So we updated it in 2023 as part of a review by Diego Morgadi. And in that updated meta-analysis, there was a tendency with a probability of 0.11 towards an improvement in feed efficiency of growth, not in milk production. So that's what I would have to say on this.
Okay, thank you, Emilio. We move on to the next question because we still have a few more to answer and time is kicking. So there's a question about if there is a list of national public compensation schemes for methane mitigation actions. I think we have already answered that. There are some countries, but I don't think there is such a list anywhere. But if any of you are aware of that list, please let me know. Otherwise, we move on to the next question, which I think can be answered probably by Andrea or Peter. What is the state of emissions from additives added to silages? Or to silage-based diets, I imagine the question is about.
State of emissions from additives added to silages. So is it an inoculant? If that is meant, then my feeling, if there's an effect, it's quite often through an improvement of the silage quality, and you see indirectly perhaps a mitigation of emission intensity. It's not a direct reduction of methane emissions, what I have in mind from literature. But I'm not sure whether the question is intended that way. Peter, maybe you read the question differently.
Yeah. The way I interpret the question is that I can say that we did a trial where we, where we fed we used the Bovaer to diets with grass and maize silage, and we tried to make those diets for grass and maize with the same nutritional composition in order to test the Kibreab equation, which is based on nutrient composition.
Yeah.
And we could see, despite that, we could still see a difference between grass and maize, a higher reduction potential in maize. So we have the Kibreab equation, and that's a really nice start. But there is still something we don't understand when we feed these different silages to the cows. And as I said before, when we can document the reasoning, then the farmer should also have a benefit for that. But as long as we cannot document the reasoning, why there is a difference between different types of silages, then it's very difficult to differentiate. Personally, I think it has to do with rumen structure, that when you feed a lot of maize silage, if you open the rumen cannula to a cow fed a lot of maize silage, it looks very different from a cow fed a lot of grass silage. So I think it's about distribution of the feed additive within the rumen, and also access to, for example, fiber particles and so on and so forth. So there is something about rumen, how can you say, dynamics, rumen physics that we need to look into with respect to these interactions with silage type.
Yeah, it could be volumes and passage rates, and my impression also is that digestibility, so just the rate of fermentation, the quality, the digestibility of the silage also adds to variation in this efficacy.
Yeah.
Thank you both. There is another question coming from Sub-Saharan Africa about the impact of the efficacy on grazing systems of feed additives. I think that has been answered already. There is a lack of studies. There is lots of research going on, but this is an area that needs a further development, and that was clearly stated in the Hegarty et al. Report in 2021. There is another question on whether there is a relation between enteric methane emissions to the age of the animal. So how do they relate, if there is any study on that? So if any of the colleagues that work with dairy cattle, maybe that obviously age is an important element, can comment on that.
Yeah, with parity, you see animals consuming more feed, you see more methane emission, that's obvious. And the more the animals feed, normally the amount of methane you get per kilogram of feed dry matter goes a bit down, but it's marginal, but it goes a bit down instead of up. But the total amount, of course, will go up with age of the animal.
Yeah, and just to add on that, we have when we look on the studies with feed additives, there's a lot of focus on the concentration of a given feed additive, you know, grams or milligrams per kg of dry matter intake. What there's not been that much focus on is actually the amount of feed additive you give to a different animal. And there, for sure, there's a huge difference between a highly yielding dairy cow or a low yielding dairy cow due to differences in feed intake. But if we talk about that there is a difference between diet in response to feed additives, that is nothing compared to the difference in response, for example, we see between young stock, young calves, and dairy cows. There we see huge differences in the dosages that they can cope with, and also in the response in methane. But that's not really been looked into yet in a sufficient way.
Thank you, Peter. Alejandro?
Yeah, a couple of years ago, we published a paper in which we developed equations to predict methane production in sheep. And of course, the dry matter is the main driver, which explains most of the methane production. This is without feed additives. But as well, we discover that the body weight as well can be an important factor that can predict methane emissions. So we develop equations specifically for lambs and then other for sheep, because for adult sheep, because we discovered that by doing it that way, the equations were more accurate. So yeah, there are other factors that need to be considered as well. The digestibility of the diet, the rumen propionate molar proportion as well was a good indicator. So yeah, it seems that we need to make more accurate those equations by including more variables, not only dry matter intake, which is the one which has been used in most cases.
Yeah, that's a good point. Yeah, thank you, Alejandro. Right, there's a question on the impact of anti-methanogenic feed additives on milk and meat quality, and I suppose the question is not about safety but quality per se. So the nutritional characteristics of the product. My understanding is that the ones that are regulated, approved, don't have a direct impact. But if any of the colleagues that have been running in vivo trials in dairy cattle or beef cattle can make a further comment, you're very welcome to do so. Otherwise, we will move on. Oh yeah, Andre?
Yeah, you do see some changes in the fermentation profile, and that obviously will also affect fatty acids that end up in the mammary gland. So you do see these characteristic changes in fatty acid profile of your milk fat. But it's not huge, but it's with 3-NOP or with nitrate, I think with 3-NOP it's even most clear. You do see always the same kind of changes occurring. Yeah.
The impact on milk fat or the fatty acid profile, is it? Yeah. I suppose it's more on the content of that fat rather than the fatty acid profile, I suppose.
No, it's just the profile. So the profile, the different types of fatty acids you get. You see certain fatty acids coming up and others going a bit down relatively. So it's quite characteristic. You do see some small changes, but and protein and lactose content, I don't expect a lot. Unless your whole energy, metabolism, feed intake levels are changing. Of course, milk changes as well. That's normal.
Okay, thank you, Andre. There is a question on, most of the feed additives being tested in mature adult animals, so in which the rumen microbial ecosystem and the rumen itself is fully developed. And the question is how much has been done in feeding feed additives to young ruminants when the rumen is being developed. I can comment on that quickly, and then maybe Alejandro can also provide some insight because he's been also working in that area. There are some works using different feed additives. We initially published a work on using bromochloromethane and just as a model, and the impact seems to, when you feed a feed additive and you stop the treatment, the impact persists, but it dilutes over time, and it doesn't seem to last for very long. But there is also kind of confusion in other works done with in one work done with 3-NOP, where the effect lasted for 2 or 3 months, I think. Work done in INRAE, and I'm sure there is all other works that are being done in other parts of the world, but I'm not aware of any other way. There is some work as well using medium chain fatty acids, but I think the overall conclusion is that you can modify the microbiota, but the effect does not persist for very long. I don't know, Alejandro, if you want to comment on that.
Yeah, I agree with you that when we do early life nutritional interventions, normally in animals with artificial lactation, we encourage the rumen microbiota to accelerate the colonization of the rumen. And that can be done relatively easy by, for example, inoculating with rumen fluid from adult animals. So it's easy to increase the biodiversity of the microbiota. But with the feed additives, what we do is normally the opposite. We want to make the rumen a bit more simple, or to remove the methanogens or things like that. And that is more difficult because, as soon as we stop the treatment, the effects tends to disappear. And it depends as well on how we manage the animals. If we keep them in isolation, probably the effects can last a bit longer. But if we mix animals from the control and the treated animals, the differences tend to disappear, because probably there is a cross-contamination between the microbes from one animal to another.
Thank you, Alejandro. So yeah, I think we have passed 15 minutes of the predicted duration of the webinar. As we said at the beginning, we will provide the content and the video and the answers to those that haven't been addressed, only a few. And I'll pass the word to, I thank everyone for the questions and obviously my colleagues for the answers. And I pass the word to Andre to close the webinar.
Yes, I don't have a lot to add. Thank you, David. I think it was a good session. Again, I emphasize, please, if you have further questions or you want to have further insight into details on these papers or to discuss something, you can find us by our emails. The work will be put on the website as well. There will also be some dissemination activities in the rest of this year. This work will be presented at the ATSA meeting, the annual meeting of the Dairy Science Association in the United States. It will be presented at the EAAP, which will be in Innsbruck in Austria this year. And it will also be communicated at the GGAA, which is in Kenya this year. So at several conferences, we will go into much more depth and detail in presenting the outcomes of this work. And I hope I will see most of you there during these workshops if you are attending those meetings. And we will soon also disseminate it to the Global Dairy Platform, who actually funded our the person who helped us, David and me, most, which is Florencia Garcia. She did a lot of work and she helped us through this whole project. So many thanks to her. She was funded by the GDP. So quite soon the GDP will have a meeting and we will give a presentation of the work at that meeting as well.
Maybe Flor can turn the camera on so you can put a face on.
Yes, you can see Flor then. Yes, so this is Flor. She helped us a lot, and actually the project passed already, but she's still helping us, which is really good. Yeah, thank you, Flor. And I think with this, we come to the end of the webinar. Wonderful that you all attended, and hopefully we can meet or discuss further in the near future. Thank you all.